Brunswick, Canada, to give this lecture this afternoon. Corey gave a talk at the 100th anniversary of the Ecological Society in America that I saw 2 years ago. And it was one of those talks, if you've been involved in salmon restoration for a number of years, you sort of stood up and did a double take. You just kind of, I really couldn't believe what I was hearing, because if you look at the challenges to the recovery of Atlantic salmon fisheries in this hemisphere, you see that how many issues that we've had to deal with damming, of excessive logging and habitat destruction, of climate change, of concerns about hatcheries, concerns about aquaculture, scabies, in the midst of a massive growth of the salmon markets in this area. So with that level of controversies, for somebody to sort of come into a talk in front of literally hundreds of ecologists and said he might have some commonsensical, rational ways of explaining things scientifically, and maybe even a route forward to recover, maybe not in my life, but in your life, the Atlantic salmon fisheries. So Corey is going to talk today, as you can see, about his work in the northern Bay of Fundy. But his work absolutely goes to the core of marine conservation and a whole movement in the world today called rewilding, and using a lot of our brain power in order to recover populations of just about any wild species out there. So Corey comes to us, as you can see, he's a project coordinator and a scientist in the national parks of Canada. He comes to us from a small village right there at Fundy National Park. Yes, the place of the 50-foot tides. Yes, the place where North America cracked apart, and I had a chance to go visit him, where I picked up rocks that in one rock it had North America, Africa, and South America in one rock. So a really remarkable place. He got his undergraduate degree from the University of New Brunswick in Fredericton, originally in forestry. He spent a lot of time here in New England running around thinking he was going to be a forester. And then did his master's degree at Memorial University in Newfoundland. So now he's going to tell us about this kind of remarkable activity called salmon recovery in Bay of Fundy National Park. Welcome, Corey.
That's quite an introduction. I'll do my best to live up to that. But I'd start off by saying I truly appreciate the opportunity and the audience. Part of my job with Parks Canada is communicating what we're doing to the rest of Canada, but really in search of excellence in conservation programs that we operate. Communicating to an international audience is extremely important. So I'm very happy to do this, and I assure you, because it's already happened, some of the discussions that we will have on this trip will improve this project, and that's why I'm so excited to do it. So if I do a good job over the next half an hour, you'll understand well where we started in our program and our problem in the Bay of Fundy, and where we are today, and why there might be some hope for the future because of it. I like this slide to start with, actually. 2003, we released our first fish into Fundy National Park, and there's a small group up there, and they're made up of Parks Canada, so a government agency, Department of Fisheries and Oceans of Canada, a government department, and Fort Folly First Nations, a local First Nations group that have sort of partnered in salmon recovery with us since the start. And fast forward a little over a decade, there's about a dozen different organizations in that picture, from government departments, private industry, and First Nations. So I suspect some of the success that we're achieving has to do with the size of that group and the different perspectives that they bring to the program. Where are we? You know where we are, I guess, but where am I normally? At the head of the Bay of Fundy there at the red dot is Fundy National Park. And we have two rivers in the park. One is called Upper Salmon, one is called Point Wolf, and I'm only pointing that out because they'll come up in the talk a few times later. Right at the head of the bay, I think it's important to note, because some more current work is happening there. This is the Petitcodiac River, and that's the largest river in the inner bay of Fundy salmon's population. I should point that out too. This is the St. John River right here. The city of St. John, New Brunswick is right here. And basically all of the rivers up and around the bay from there over to here are called the inner bay of Fundy Atlantic Salmon River. So we're talking about 40 rivers that used to have a population of approximately 40,000 returning adults. And by the mid-90s, that number was likely less than 200. Today, where Atlantic salmon occur in those 40 rivers of the inner bay of Fundy are really solely a result of restoration work. The majority of those rivers are extirpated. That's basically what the coastline looks like. It's not terribly different than some of the places around here. And the last thing I would note, it's important to point out where Grand Manan Island is. For those who are not familiar, that's a really key part of what we're working with today. Marine survival. So for those who don't know the salmon life cycle, in the fall, fish would come back to rivers. They would find a spot where they would dig a hole in the riverbed. They would deposit some eggs. The eggs would grow in the river for a couple of years as small fish, and then they would leave the river as what we would call smolts when they're approximately 3 years old. And it's at that stage that the cycle seems to have broken for this population. They fail to return from the ocean. Normally, inner bay of Fundy fish, what makes them different, they would leave those rivers from the inner bay, and they would migrate down to the mouth of the bay and Gulf of Maine. They would spend a winter, and then they would come back to their rivers to spawn. It's somewhere between their rivers and down here, they're not making it back, and that's generally the consensus for what is limiting the recovery of this population. So in the early 2000s, we find out that indeed this population is at risk of disappearing altogether, especially from our park rivers, and we go out into the park and we find out, well, what's happening? How are the fish doing in our park? We find that there are no returning adults, and there are only a few and declining numbers of juveniles present in the river. So the response at that point is to collect a sample of those juveniles from the river, because as I just explained, they're kind of on a doomsday course. They're going to grow up in the river, they're going to leave, and they're not going to come back. There's no adults coming back to make new ones. So extirpation is really imminent. And that's really what we did. We went to the river, we collected a portion of juveniles with a rig like this. It's called a smolt wheel to most, a rotary screw trap to some, and that's the device that collected the remnant population that we started our work with. This is early 2000s now. They went to a Department of Fisheries and Oceans hatchery, not unlike most hatcheries that you know about, where they were grown to maturity and genotypically kind of identified. And then we would spawn them in captivity and release them back to the wild as either fry or par. So it's important to see those two. That's what they look like. Fry are simply fish that have just hatched out of the egg and they're ready to feed on their own. Par are the same fish, just held at the hatchery for an additional few months and fed in captivity and then released to the wild. So that's the difference between fry and par. And depending on which family the individuals were from, sometimes we would release them as adults. If we already had a lot of copies of that individual, we could release it just back to the wild as an adult. So what this results in, this is an important part so that we can go forward understanding and you get the most out of this that you can, is that we collected some wild remnants, we held them for a little while in captivity, and we put them back into the wild at different stages. And that cycle generally repeated for quite a while. And what we found, Parks Canada is responsible for monitoring the ecosystems of national parks across the country, and Fundy is no exception. So monitoring operations on our rivers, primarily at the smolt wheel and a few other methods, showed that smolts leaving the river as a result of those three different strategies looked different. And this really started, this is, monitoring. We like to make the analogy of monitoring in Parks Canada to like your family doctor who takes some kind of like boring readings time and time again, like blood pressure and heart rate and weight. And from those really boring measures, he can infer when there could be a problem. And that's sort of what has happened here. So we're just taking lengths and weights and timing of migrating smolts, and all of a sudden things are pointing to these fish are different, and why, and could that potentially be affecting their ability to survive in the ocean, which seems to be limiting the population. How do you find that out when none of them come back from the ocean? You don't know. You know they look different. That might be having an effect on their ability to survive in the Bay of Fundy when they leave, but none come back. So the answer is we don't know which one might be the better strategy, because none of them produce a return. So in 2008, 2009, this started a project. We approached the aquaculture industry operating in the southwest of New Brunswick. They're growing millions of adult salmon, and we have none. So they were a reasonable group to talk to about this. And we more or less asked, when we catch some smolts coming out from these different release strategies, would you grow them to adults for us so that we can see what's going on? So we released those two different strategies into one river, so just concentrate on fry and par for now. A few years later, so we released them in 2008-9, 2010 and forward, we're catching them at Smolt Wheel, and you know, you don't have to be a Philadelphia lawyer to see that those fish look a little different, even from the back of the room. So those are smolts that are leaving the river as a result of two different release strategies. So everything else is the same. They could be brother and sister, in fact, but they're quite different. And the only difference in their handling is whether they were released to the wild as a fry or as a par. From there, this is where the kind of new partnership starts in the southwest corner of New Brunswick, where all of our salmon aquaculture is occurring. We take them to specialized marine pens. The industry steps forward, develops different, very small salmon rearing pens for us, and we grow these wild fish on domestic sea farms. And when they mature, we monitor them the whole way along while they were there, collecting different measures. But the most important one is at the bottom. When they matured, we brought them back to the Department of Fisheries and Oceans hatchery, and we spawned them in captivity there. And more or less, we spawned groups of fish that originated as fry and groups that originated as par, and we produced eggs, little baskets full of eggs, and we monitored their viability going forward. And one important thing to note is we released all of the adult fish at that point in 2011 back into the wild, and thanked them for their contribution to our efforts. Key results are published in this paper, 2016 Animal Conservation, and overwhelmingly, whether you can see or not, reduced captive exposure, RCE, and extended captive exposure, that's fry and par. But reduced captive exposure made older smolts, heavier smolts, slower growing post smolts, but that sort of fits with the way that small fish grow at a faster rate than large fish. Larger adults, larger eggs, and more viable eggs. That's really the most important one, the very bottom one. So a few more months spent in the wild had all of those effects. We're talking a difference of like 4 or 5 months for these individuals. So keep the fish in the hatchery for another few months, and you lose 5 out of 6 of those measures at least. You can add this up in general to the less time these individuals spend in captivity, the more measures of wild fitness they sort of portray. And if you follow the logic at this point, we're going to make an important leap or step depending on how with me you are. But adult releases produce fish with no captive exposure. So if less captive exposure makes more wild fitness, you know, less captivity is better. Wouldn't no captivity be best? And that's where we went forward with a desire to catch juvenile fish, make them into adult fish, and release them back to the wild so that mate choice and the timing of spawning and the location of spawning and all of those things, all of those early exposure attributes for this animal are happening in the wild, free from any influence of captivity or artificial environments. Now, how would you release a lot of adults if that's what you wanted to do? It's not an easy thing to do with a hatchery because it's expensive and it takes a long time to grow big salmon. but in this early research, we discovered a partner who was not only dedicated, but extremely capable of growing adult salmon to the tune of millions. So they were a reasonable partner to approach. So before I go to the next slide, we're going to back up because there's a few more results that I'd like to touch on because they are quite interesting, but not yet published. Remember, we released those fish at the very end down there, and that was in 2011. Well, in 2012, we hoped that the 2011 fish would spawn, these great offspring, really. But they themselves survived in numbers that, were really quite impressive. Added up to, approximately 50 in total, 42 salmon observed in 2012. That constituted like a 25-year high for that river. So it doesn't sound like a lot, but it made the 6 o'clock news kind of thing. And but all of those fish were released the year before in 2011, and so that was something, I guess. And following this forward, 2012 we had these good returns. They hopefully spawned. Well, in 2015, their smolts would have been leaving the river if they survived. And sure enough, so we had these kind of record numbers. Both of them were record, 42 and 13 were like 20-year highs on their respective rivers. And remember early in the talk, I started by saying sometimes we would release adults in the early parts of the program. We would release them in one river. So we have some information from early on that was just more like a byproduct. We have to release these adults eventually as part of the live gene bank program. And we developed some fairly interesting correlations that I would put up there and just say that we have some idea how many smolts to expect with a given number of adults released. Now I know that's very rough and dirty kind of comparison because sex ratios change as maturity ratios change and everything like this. But in general, look from 2003 to 2010. Basically, we would catch often a smolt for every adult we release, and that we catch about 10% of smolts. So what we're saying there is, in very general terms, and a released adult might produce 10 smolts in total. And here's the number of smolts that we saw in 2015 coming out of both of those rivers. So we observed 42 salmon. On the old scale, we would have expected to catch 42 smolts, and we caught 700. Similar situation on the point wolf. It's important to note, and I think I point out in other places in the talk, in 2012 on the Upper Salmon River, we did release a few other fish. They were released at the mouth. So in effect, there could have been more than 42 individuals contributing to the parentage of the 2015 smolts. And we do have some more information on that to say that it is the case. But on the Point Wolf River, for example, there were no other fish released. It was just those 13, and we caught 10 times more fish than we would have expected from 13 individuals. All of this points toward our intention that those adults that returned in 2012 had more wild exposure than any adults we've ever released, and they had very good surviving offspring. So everything's sort of in line here. Those good surviving offspring come out of the river. We catch them, and we're very interested to know how related they are to those adults that came back here in 2012. So we captured 29 of these individuals that came back, and we took a little piece from them, thinking that they would be spawners. When the smolts leave the river, we take a piece from every one that we capture, and we link this back to these parents. And the overall results are right there in the middle. The spawning success was quite variable, so we had only 29 samples to compare with our 700 and some smolts that we captured. But some things did come out of it. We found only 16 of 29, so half of them spawned. So that might be surprising to an individual who's thinking that if I put mature fish in the river, they're all going to spawn. So that was interesting. One thing that did come out of this, though, is how many females produced apparently very good surviving offspring. So in nature, it wouldn't be uncommon to see 1 or 2% of eggs laid make it to the smolt stage. And 10 out of the 12 females produced 50 estimated migrating smolts. So 10 different times that happened. That would be comparable to wild numbers. That's about how many eggs these females would have been carrying. 4 of them produced 140. So 4 of them did extremely better than we would see in most situations. And 1 of them just kind of, went right off the charts and made 740. So this is likely an effect when you stock an empty system, the individuals do better because there's very little competition. So there's likely something like that happening. But all of this is to say that the offspring are apparently surviving very well in many cases. So the last part of all of this results thing, so there was the published results, then the fish came back, and then their offspring went out. Did those offspring come back as returning adults? And this is all from the experimental phase of this work, which was supposed to end with the publication. We didn't see with our dive surveys any returning adults, but we have other mechanisms to detect them. And our sonar is set up at the mouth of the river that detects all fish going by, whether they are wild or released. And information from that system suggests that up to 8 individuals did come back into the river that weren't wearing a tag, which could mean they were wild. It could mean other things too. Back to less captivity equals more wild fitness and our desire. So now we've got even more information. We really want to focus on releasing adults. How are we going to do that? We go back to the aquaculture industry and we say, hey, we got a really encouraging kind of spark from this experiment that we did. Would you be interested in growing more wild juveniles for us so that we can see whether this is a fluke or what else we can do with it? And they said absolutely. And from the experimental version, we think that the thing that you would want to do here is have a dedicated farm, and we'll work to help establish that. So Cook Aquaculture approaches the village of Grand Manan, who own like a community garden, but it's an aquaculture garden. And they establish this farm in Grand Manan that looks in many ways like a domestic salmon farm, but it only has wild salmon on it. And this is the first one of these established sites that anybody has heard of that I've talked to in the world. So we've kind of put our feelers far and wide for this at the World Aquaculture Conference. We don't know of any examples of this happening elsewhere. So basically what is happening right now, starting from 2014, captive hatched individuals that we've already gone through, like at the hatchery, they're released as fry to the wild. They're collected later from the wild, so we're exposing them to as much wild as we can in these circumstances. We catch them as smolts. They go to this site, so they're rearing in the Bay of Fundy to adult, and then they leave that site. Starting in 2015, we've been releasing adult salmon into the park rivers in kind of significant amounts. And that's sort of what it looks like there on the right. We do most of this with Coast Guard helicopters because the rivers are so remote. And in 2018, we will start catching smolts from the first released adults. And those are going to be special smolts because those smolts will have been hatched in the wild, never exposed to captivity. And if you can fast forward in your head to see what does this actually look like down the road, we'll take that smolt in 2018, we'll take some of them to this site, we'll grow them to adults, they'll go back to the river, and then we're going to catch smolts in the next generation that are special again because not only were they hatched in the wild, but mom and dad were hatched in the wild. And domestication research would suggest that, if you were hatched in captivity, you're less wild than your parents were, and if you have babies in captivity, they're even less. So you get really bad at living in the wild really fast. Can it work in reverse? We don't know. Here's what it looks like though. So this might not mean a lot to many, but for those who have been directly involved in salmon conservation work, like myself for a long time, I've looked at this same pool for 15 years, and not counted more than 15 fish in grand total. And in 2016, there was probably 300 in there. over 840 in the whole river. So this is something that is really kind of turning some heads. This river is kind of full of fish like it was 50 years ago, 60 years ago, or even longer. This river would normally have, its record high count is 1,200 salmon. So we were close to that in 2016, and we're likely to exceed it this coming fall. So to sum it all up, diverse collaboration is required. You know, our First Nations partners, Fisheries and Oceans and Parks Canada, kind of look after getting the fish from the wild. Department of Fisheries and Oceans handles them in indoor captivity situations, making sure that we don't lose our unique strains of Atlantic salmon. And what is not needed for that live gene banking program is funneled to the wild salmon conservation farm operated by Cook Aquaculture, and other partners are needed to make that go forward. But all of that is adding up to unprecedented numbers of Atlantic salmon going back into these rivers that haven't seen them for decades. And our hope is that they're going to produce great offspring. So diversity in partners adds up to some results, and those results are allowing stuff to happen also, because these are kind of unprecedented times, right? What is happening when all of these salmon are in rivers? Well, there's lots of research going on to look directly at that. UNB is looking at the effect of all of these adults are bringing marine nutrients like fertilizer into the river. And they're actually showing that that fertilizer is so important that when the river has these adults coming back to it, or being placed in it even, there's more slime growing on rocks, and more slime means more bugs, and more bugs means more food if you're a baby fish, and more food means more baby fish are leaving the river. And this is a real feedback type of effect. We have habitat research underway at under Dalhousie University with Dr. John Grant and his students, really not possible in many other rivers in the Bay of Fundy because they have no salmon. Surveying habitat is one thing, but surveying habitat and showing how well your predicted habitat is lining up with where fish are spawning is a far more powerful thing. They're using a drone to collect this. I'll show you. It's kind of a whiz-bang thing, but pretty interesting opportunity because fish are in the river. And the same thing with counting these fish with a sonar. So I'll show you a little bit more. This is the marine nutrient stuff, more or less. Put these fish in, you see a surge in nutrients. This is actually from another paper, but it's a better example than the early results that we have. But you're seeing an impact already in Fundy National Park with the input of these nutrients. Nutrients are staying in the system longer, which is akin to there being food available for you to eat for a longer period into the winter. The tracking of these fish is another important thing. So we release these adults to the river. They all have tags. We want to know where they're going, if they come back in the future. We track them in a couple of different ways. One way is passively. So at the base of the river, that red line represents an electric gate. There's a cable running under the water that, just like scanning your bag of chips, when the fish go over it, they have a pit tag in them and it beeps at the gate and gives us a record. Otherwise, all of the portion of the fish are wearing, about 50 of them are wearing like a radio tag. And this is the same technology that you would track like bears or deer or something like that with. And we have some very keen technicians who go up and down the banks of the river, and they locate every day that the fish, or every week that the fish are released, where they are in the river, and that's what those red dots represent. And so just remember that every fish is wearing a tag, and they're detected when they leave the river. Some of them have fancy tags, and we detect exactly where those fish are every week. Here's what some of the habitat data look like that's been collected with the drone. So this is three-dimensional spatial data for anybody who has any kind of GIS experience. So this is not just a pretty picture, it's real spatial data. Every pixel has information attached to it, and it's going to allow us to, when we lay the tracking data over top of this, you're going to see what habitat characteristics are associated with where fish are spawning. And because this is collected with a drone, it's a bit of a kind of a neat thing that I don't know very much about. These are the experts here that are doing it. But basically, you'd be able to fly this another river with our model loaded in it, and you would get an estimated, these are your spawning areas, these are your holding areas, and so on, which could help river groups understand more about where they should put fish if they were going to release them, or where their spawning habitat might be. This is just kind of a sample, I guess, of the data. Hopefully I can get that to go. Yeah. So you can see the detail even, the guys would be really mad at me because my computer doesn't even have enough power to show the real detail, but this is what the drone is collecting with a spatial camera, and this allows kind of an unprecedented characterization of the river to occur. This is early days, but the detail is amazing. Essentially, they'll be able to give me a count of the area where the pebbles are less than an inch kind of thing, or between 2 and 5 inches, and then you'll have a whole map laid out that shows you where good spawning habitat could exist and so on. Another thing, sort of unprecedented again, because we actually have fish to count. There's really cool ways to count them, and this camera exists. This is a Didson sonar camera, and I'll explain it a little bit, but it exists below our pit tag array, that electric gate. So theoretically, if you swim over the electric gate and you're not detected, that means you're not wearing a tag. And if you swim by the Didson sonar, you are detected. So the difference in count between the Didson sonar and the electric gate should theoretically be your wild fish count, right? The Didson saw 10, the electric gate saw 5, so that means there must have been 5 fish going through there without a tag. These are early days, and these systems are not without their error. Sometimes a tag can fall out, sometimes a fish can be undetected, but we're working at kind of roping that error down. And hopefully, if this works, you can see this is a little example from the sonar output of a migrating salmon, but you can't. There we go. So the cool thing about this is that it's not seeing with eyes, it's seeing with sound. So when the river is really turbid, it doesn't matter. It still detects your fish going by. What's most important for us is it works in the dark, because that's really when fish move the most. So normal cameras aren't as useful. So those are a few examples of kind of the unprecedented research that we're able to do because of these results and these fish being in the river. But what's really taking more of the focus time and time again are the non-research things. And some impressive things, some impressive collaborations and programs have been allowed to occur because of these results. And I love highlighting them, even though they're kind of non-scientific. But our park wardens in the park come to me and they say, how are we going to protect all of these salmon? We're only 2 park wardens. How am I going to make sure somebody's not taking them? Well, they figured it out, and they went to all of the local law enforcement jurisdictions. So these are like federal fisheries officers and like off-road enforcement people and the forest rangers and all different kind of jurisdictions to make sure that wherever those fish go in and outside of the park, somebody on this team has jurisdiction to look after them. Huge public interest. This is the day, just this fall past, where the fish were flown back to the river for release. The Striking Balance, check it out on tvo.com. It's a 1-hour-long documentary about the program and all its collaborations. We just had a really nice feature of the wild salmon farm in the recent issue of Canadian Geographic. The Swim with Salmon for Science program, this is a Parks Canada, like a visitor program that you would, as a visitor, just go and sign up for, and you'll get suited up with one of our biologists, like in all the same gear that those guys and girls use, and you'll be taken literally by the hand to some pretty special places in the park to snorkel and count these fish just like they do. And this is a young man who I might like to think that otherwise wouldn't have seen a fish, an Inner Bay of Fundy salmon at least, in that river if it weren't for that program. So that's the first salmon that he's ever seen, and he's only grown up like 2 hours from the park. So, and of course, and last but certainly not least, the Petitcodiac River is operated by our First Nations partners. It's the biggest river in the Inner Bay of Fundy, and now they're working the exact same model as us. They're collecting fish from the Petty, they're releasing them back as adults, they're growing them at the same marine farm. So this is how the project is starting to scale up and have an effect outside the boundaries of Parks Canada, and I'll more or less shut it off right there and leave you with, again, a thanks for the audience, and I guess, a comment that these types of collaborative programs have really been the key to us moving forward at the rate that we have. It's when all of these agencies have finally gotten together, progress has been able to be had. So thanks very much for your time.
Okay, so this is being webcast worldwide right now, including the country. So we want to take your questions using this, okay? Come on. First, Catherine, you the first three have to be, first five have to be for the students in the class.
Hey, it's loud. Do you think the tags and the radio tags affect the fish's fitness?
Affect the fish's
Fitness? And their ability to survive in the water?
the fitness,
Yeah.
right. We've been doing this for a long time. Sometimes that's not the best answer, but that provides us with a fairly deep base of evidence to say that these pit tags, the pit tag is like the size of a grain of rice. It's the same type of chip, really, that might go in your dog or cat. It's a little glass capsule. You can't, after a month, you can't see where, and we just slide it in under the skin. This is how fish are inventoried in the vast majority of hatchery type of settings. So we've got them in a captive place for 5 years to see that this has very little effect on them. So I wouldn't worry too much about the pit tags. The radio tags, they're a larger tag. That's more like the size of a AA battery. And how that tag is applied, sometimes they can be put internally, like in the belly, and then sutured up. Sometimes they can be put kind of like on a backpack on the dorsal fin. How it's applied definitely has an effect, and how the fish is handled after the tag is applied. So there's a big difference between putting this thing in with some sutures and then just kind of, putting it in the river. That's much worse. If the fish is allowed time to heal, you as a researcher have more confidence that, it's taking the tag okay, but we certainly have had instances where tags have fallen out and we have lost fish to that method. But the idea here is that the data that we get are data we don't have. We don't know where they're spawning, for example. So what we get from them really is valuable, but it definitely has an effect, the larger tags. Yeah.
What do you believe are the main reasons that fish were not returning?
I don't think I've ever gave a talk without that question. There's probably a lot of reasons. One of the ways I like to start that off is maybe some of you will be familiar with the case of the Peregrine Falcon in the Bay of Fundy area. It was near extinction, and then some smarty pants like you guys in a lab somewhere figured out that hey, there's these eggshells are too soft, and chemical analysis revealed that there's DDT in this, and that's what's keeping it from being hard. You take the DDT out, the eggshell's hard, chicks survive, peregrine's great, and they have rebounded. One of the best success stories in endangered species in our area. Salmon is not likely that simple. It's not likely take out one thing and everything will work. It's more likely the accumulation of a lot of things happening probably on the land and probably in the water, and changing or stopping or modifying any one of them is unlikely to affect a lot. The most commonly cited ones are interactions with aquaculture. The timing of the collapse of our population coincides well with the growth of the aquaculture industry, but coincides well with the decline of cod stocks too, which are a long way away from the aquaculture industry. Damming of rivers is something that we love to do. In 1968, we dammed the St. John River and the Petitcodiac River, the two largest rivers going into the Bay of Fundy. I don't know, it was a particularly hateful year, I guess, but this stuff seems to happen far and wide. The impacts of all industry, forestry, farming, they all have had kind of argued effects, but I'm certainly not going to offer any opinion on which one might be the worst. It's likely a cumulative effect. In our case in the Bay of Fundy, my own opinion is that it's increasingly important that groups like yourselves are actually interested that there's a problem. That's the first step to finding out a solution. If nobody cares about it, they're not going to recover themselves. But another thing is the population numbers are so low. Imagine a pipe that has no water in it, but it has a leak. How do you find that leak? So, you search miles of pipe, but if you can run some water through it, you can pretty quickly see a leak. So I think there's so few fish right now in the Bay of Fundy that one thing that this work is doing is providing researchers with more opportunities to track larger numbers of fish and see exactly where they are going and what is happening to them or what could be happening to them.
Are native fish having any real contribution to the populations you guys are looking at, or is it almost entirely a result of your supplemental stocking?
Where we are in Fundy, so in the first couple of years of the program, we collected what you mean in that case, that would be native fish under what you're saying, because we didn't put them there. Those fish were very likely strayed from other rivers. These are the last stragglers, of a population. So those are native fish, and we collected remnants for as long as we could. As long as they were there to collect, we would collect them. So that added up to be two years, and then it actually went to zero. There was a smolt run that we set up, and we did a whole trapping campaign on, and not one fish was caught. So that might have suggested that we got the last few. That was it. They trickled out and they were gone. And then we've built all of these fish, really, from those early stocks. It is very unlikely that there's much, if any, what you might call native fish coming to our rivers right now. The fish that we are seeing there are either put there directly by us or put there in the past by us, or the offspring of fish that we have put there. Without this work, I think in 2006, it's estimated that population would have been extinct. Yeah.
Are there different methods in raising salmon to adulthood in captivity for these efforts versus raising salmon to adulthood for food fish?
Yeah, absolutely. So what's the difference between how we grow fish at the wild salmon farm versus at the other salmon farms? So the answer is absolutely. And it's generally to do with the number. These industries are set up like most industries are to produce the product the most efficient way that they can produce them. A feed blower that feeds 20,000 fish, you turn it on for like 1 second to feed the fish that we have in these cages, because we're talking about hundreds or maybe up to 1,000 sort of thing, but definitely not tens of thousands of fish. So the cages are much smaller, so the practices to feed and care for those fish are different. They are more manual, more labor intensive. The farmers actually spend more time over top of the fish looking at how they're feeding and how they're performing. So it really comes down, I think, to the number of fish that we're growing that affects it. But they are, innovating new ideas and methods all the time at this place. They're talking about more custom diets that might better mimic a wild diet for these fish. We're really trying to grow mature fish in this model, and that's really not what domestic fish farmers want to do. They want to grow large fish, and when fish mature, they generally stop or slow in growth. So mature fish is generally something that the domestic farmers want to avoid. Mature fish is our goal explicitly in our model. So the techniques do change a little bit. They're largely based on the numbers, but new products and innovations have definitely been developed and are still being developed to grow wild fish best.
You had mentioned that there's the increased slime, so there's more bugs in the river, but are there any other effects on other species that aren't necessarily fish in the river as a result of the work that you're doing, or like before the work that you were doing?
So this work is being led by a postdoc at the University of New Brunswick, so I should make very clear that this is his work. His name is Kurt Samways, and his PhD work and master's work both focused on these types of food-wet interactions, and suggest that there's a broad effect. That's really the whole purpose. It's not just for fish; it's for the entire ecosystem. It's measured in different ways, but I would draw your attention to more West Coast examples for this. The West Coast salmon, which die after they spawn, have a very obvious and large volume effect on that ecosystem, and that definitely affects things like bear survival and trees. They find them even in the rings of trees, these nutrients that are brought back by the salmon. So if our systems are comparable in any way to those, we expect that the effects are broad. The measurements that Kurt is looking at are really on invertebrates and fish, but one could assume if there's more bugs, there's more food for a lot of things. If there's more fish, there's more food for a lot of things. So it is likely having an effect further up the food chain than even we are measuring.
Anybody else? No other questions? Yes?
So as the waters continue to warm in our area, how do you feel that that's going to impact salmon's viability?
Specifically marine or freshwater or both? Are you thinking more in a marine context or more in the river?
I guess in the river.
Yeah. That's a good question, particularly in my context, because our aquatic systems are likely sheltered from a lot of the effects that other aquatic systems are experiencing. Have a look at Fundy National Park on Google Earth, and you don't need to see the outline of it because you can see it. It's where the trees are. And outside the park you can see it is more industrial forest. So more open watersheds are likely going to have an effect downstream of warmer water. So we may not see that. We don't do those comparisons to say that that's occurring, but we might be isolated from that a little bit. The effects of rising temperatures or high temperatures on salmonids are well documented and negative. It will be a bad thing, and that could also explain the latitudinal gradient that seems to be occurring with the occurrence or the disappearance of salmon. Seems to be a line below which there are generally no salmon in North America, and above which there are still populations that are intact to various degrees. So warming is certainly on one of the other questions. It's on the list of culprits, and if it is the culprit, obviously a very difficult one to address. Not an easy question.
It's interesting though. I worked in salmon in Southern California, where there was a strain there that did quite well at 23 degrees C. And on this coast, they used to be down as far as the Hudson River.
Right.
So they are very plastic in terms of their temperature tolerances, much more plastic than people give them credit for.
Yeah, and I mean, to further on that point, I've seen similar research with pH. Salmon are one of those things, salmonids maybe more in general, that they act this way except for when they don't. And, there's an example of almost, they migrate at 2 years old except for when they do it at 7, and they come back in the fall except for when they come back in the winter. So it's, there's a lot of exceptions to everything, and it's a very kind of plastic kind of life history for this animal.
Any other questions? John?
On the drone survey in the river, is John going to try to parlay that into like a carrying capacity model?
The initial interest in that work would be to really evaluate the efficacy of characterizing the landscape in general. John is not necessarily a fish ecologist, but he would use his skill and his lab skill to say that it was really quite fascinating to see where he did it. And again, they are the experts on this, not me. I'm just presenting their work. I don't always like doing that. But they lay a grid over the river, and then the drone flies the grid. So if we can use that model to characterize something like substrate size, then we take that to the fish ecologist and we say, here's what the substrate size looks like characterized across the whole river. And the ecologist says, well, here's where all the fish were. So we're seeing links between this type of substrate and fish spawning. And what John, I hope, would then be able to do with that is say, here's a great example of this tool in action that could be applied on another river, but it could just as well be applied in other landscapes or potentially other aquaculture applications as well with his files.
How long do you think you'll be able to see these numbers increase, and what impact do you think it's going to have on, I guess, the economy in the area?
What do you mean economy impacts? Like
So-
fishing or something?
Yeah, so if you have stocks return to, what was it,
The heyday,
4,000?
yeah.
Do you think that salmon fishing will return to the area and that will create another source of income for fishers?
Great question. Maybe the best question, or my favorite one anyway. This is an endangered population of salmon. So we are not close to having a fishery for an endangered population. But that your thoughts went in that direction are interesting, because there is a lot of interest from other groups that are looking at populations that are not endangered. Populations that, to put it generally, that if we collect some little ones and put some big ones back tomorrow, the next day you could go fishing. And that is exciting, and that is one of the reasons that these groups are approaching us for information on how we're doing this and what we're finding and problems and so on. So if we can demonstrate that this is effective at producing as fit of an individual as we can, it could open the door for more groups pursuing this for that exact reason. I think we're quite a ways away from this in the Bay of Fundy, but other populations that are in kind of less dire straits, where they're talking about, we're down to our last 20 fish and we should have 500. Could I just collect some, they're still producing thousands of migrating juveniles. Could we just get some of those and go back to where we were 20 years ago? These discussions are happening right now, and we might see them for that exact reason, to improve not only the ecology of the river, but the economic situations around, they're missing that economic driver too.
Did you run into any problems with competition with like brook trout with the young salmon? Like did their populations grow in the absence before you guys started restocking the fish?
The brook trout, Fundy National Park, like many of the rivers of the inner Bay of Fundy, are cold and they are clear and they are rocky. One of the easiest places in the world to be a fish biologist, because it's a brook trout, it's an American eel, or it's a salmon, and there's not very many salmon. So, so we can see very well the effects of these things, and in general, there's no boom of population of brook trout. And I think if Kurt were here, he would go on a real tangent with you, because he would say that the lack of those marine nutrients coming back to the river means that there's a lot less food on the menu, and that means even though there's no salmon there, it's not. He describes the system sometimes as too sterile, that there should be darker water because there should be more nutrients, and part of that equation could be endogenous fish bringing nutrients from the ocean. So there's not a great population of brook trout really before or after our efforts to this point. We haven't seen a huge change.
Is there a variation of the weight of the salmon once they return to the river, or are they all relatively normal?
So salmon in our area are described as grilse, if they spend one winter at sea. So they leave the river in the spring, and then they come back to the river the following fall after spending one winter. In rivers like the St. John River and further down into the States here, most of those populations would be characterized as a 2-sea winter population. So they would spend 2 winters generally feeding off of Greenland by most current understandings, and they would come back as what you would call a multi-sea winter or a large salmon. In the Fundy region, at least, there's pretty much just those 2 sizes. Some of those multi-sea winter fish, if they were to come back a few times, which obviously I haven't seen very much of in my time in Fundy because they've been largely gone, they can get bigger and bigger and bigger, but basically salmon are called small salmon and large salmon. The small ones can be, let's say, 4 to 6 pounds, and the large ones might be 10 and above, and we've all seen the pictures and stories of larger 15 or 20 pound fish, but it's basically one or the other, small and large salmon. Small ones are 4 to 6 pounds for the most part as gross.
Corey, thank you so much for coming, giving us great hope for the restoration of the King of Fish.
Yeah.
Thank you for listening with